Okay, done. Okay,
Yeah.
great.
All right. Recording
All
in
right.
progress.
Yeah.
Great.
Okay, done.
All right. So I'm going to do the full screen mode. So you guys can see it. All right. So the title that I gave was Tamso Ma Jyotirgamay. That is very pertinent to the work that I have been doing for last 30 plus years. Since ancient times, we know in our culture, scriptures, we always want to go from darkness to light. And that's a very general human phenomena. We like to go to light. From agyan to gyan is one way, but darkness to light. How do you see light? What is light? What is the perception of light? Vision is really extremely important in human beings, and light is what creates the vision, right? So if there's no light, you cannot see anything. So that's what we want to go to. So my work in the last 35 or so years has focused on how do we see and the cells that allow us to see. And those cells are photoreceptors. Actually, whether we believe it or not, most of us don't know that photoreception is one of the ancient process during evolution. Even some of the single cell organisms had this photoreception that was used for a sort of movement, such as the case in Euglena. So it has evolved for us to sort of recognize different objects and space and motion, all the things actually that we do, even the higher order functions that our mind or brain that we were talking about earlier perceives, they all come through vision, most of it at least. Vision is probably the most important of 5 senses that we have. So I think it's sort of, it's not hard for me to convince this group that if loss of vision is probably the most scary thing besides cancer. So what I would like to do today is tell you a little bit of our work, and rather than going into details, I'm just going to give you a brief sort of overview of what I've been doing on genes, networks, variants, and the last word, karma. How come karma fits in all the genes and networks? So I spent a big part of my life in Michigan, and I'm a big fan of Michigan football. Just imagine, just look at this particular sort of field. This is football field at University of Michigan. You see literally tens of thousands of people around of different color, and you have people in different shapes, different sizes. There's a depth in here. People will be moving around. Your vision, your eyes can really look at everything, perceive everything in real time, and that send that information to brain. But think about it, all of this information is being captured by simple cells called photoreceptors. And there's a binary sort of code either on and off of those channels that are sending the signals. All of this information is being sent, and this information is not only captured in the retina. So photoreceptors are in the retina, which is at the back of our eye, and sort of appropriately is called the window to the world. Now, in the retina, the photoreceptors capture this light information, and then there are many, many different kinds of neurons that will integrate that information and also process to some extent and put it in certain packets of information. And that information is then sent via optic nerve to brain, where brain decodes that information and allows you to make sense of that binary information that has been sent there. So this is sort of Desmond Howard who actually scored. I mean, this is during the Michigan Notre Dame game, and my daughter was born at halftime, and we were watching that. This was the Heisman pose that he made, after that. So it's sort of fun to watch. But you can't do anything if you have impairment of your vision. Like everything else in life, like genes and proteins don't exist alone, they're part of this network. My life also has been part of my both scientific and personal network. And here is a network that I showed in one of the award lectures. And these are the people with whom I have most papers. So each of these circle is a paper, is a manuscript, and they're different people. And you can see there are sets of notes and people who have had major collaborations with me. So I have always considered, and this is written in Braille, and this is really has been always the motto of my life. This is a plaque that was given to me some 20 plus years ago, and it's always at a prominent place in my office, actually just sort of below my computer screen. And the joy is in the journey. It doesn't important whether it's not important whether you reach your goals or not. To me, always is important how you get there, and are you enjoying that journey. Everything that you do, you need to have fun. So that's really, so I want to thank my mentor. When I said network, and there's a personal network and there's a professional network, but my early mentors have been a big part of my scientific career. This is, of course, late Professor Ram Sharma, who gave me a chance to join ISC, and Professor Padiati here. And this was when I was leaving. I think this was some gift that was being given, and I'm always indebted to Ram Sharma for teaching me to have passion for science and passion for whatever you do. Ram Sharma loved mitochondria, loved H2O2. And to tell you the truth, I hated my H2O2 at that time. I wanted to clone genes. I wanted to sort of do, and we used to run to the library to get the latest issue. And this was our group. We called it 77 group. We used to get together every week and present the latest paper, and we will go to library in ISC and sort of try to see who presents the best paper, and I will always pick a paper in molecular biology. So I was just sort of nuts at that time. And I want to thank GP and KPG and Guru. We used to call him Guru, not for science, but for the first time I had beer with with him. That's why he was our Guru, Nagaraj. So I think KPG actually was the person who finally sort of connected me to Alan Garan. And I had many fellowships, very fellowships which were twice the salary of what I was getting at. Actually, 21,000 in those days was pretty good, and 18 and 16 and all. But Alan gave me 11,000, and I went there. And thanks to KPG that he recommended to Alan. That was my chance to get in genetics and Drosophila genetics. He was wonderful. He didn't sort of like, in today's environment, everybody wants to, the PI or the professor to hold their hand and teach and mentor and guide. Alan's thing was, you're a postdoc. I'm giving you a person, do on your own. The only thing he taught me was how to streak the bacteria and get single colonies, which I actually didn't even know. So Alan did that, and after that, I was on my own. And I know many times I probably in the first year, I wondered whether I made the right decision or not. But I think in retrospect turned out to be correct. Then I realized I cloned a P150 glued there, which was sort of an important gene. It turned out to be dynactin heavy chain. And later on, and that we published the first paper actually on dominant negative hypothesis. It was in the discussion, but Alan is a conservative, I mean, he just passed away last year at age of 90. He retired at 96, GP. So this is just for you to remember, age is just a number. Alan retired at 96, and 8 months later, he passed away. So So you have to be always sort of into it, you do what you like. And that's what I always say, age is just a number. So then I'm colorblind, so I didn't like Drosophila looking under the microscope and figuring out what kind of color their eyes have, blah, blah, blah. So I moved to human genetics to Uta Franke, who was just, again, an amazing mentor. And that's where I went to clone the gene for Duchenne muscular dystrophy. But within 6 months of my joining there, Lou Kunkel and Ron Wharton cloned Duchenne gene, and I thought my life is over. What do I do? That's just terrible. And if you think about it, by positional cloning Duchenne and chronic glomerular disease, this CGD, there were two genes that were discovered within the deletion that Uta had described before that in a patient. So Haren, as usual, he was the big connector, manipulator, politician. He sort of connected me to Shem Weizmann, where he was at that time, and praised me. Said, oh, this guy is good, take him and all that. So Sherman sort of said, all right, I'll give you 2 years. I said, but I want to write my own grant. He said, what do you want to write on? I said, I want to clone the gene which is next to Duchenne gene called this X-linked RP. He said, what do you know about RP? I said, I don't know anything about RP. I don't know anything about retinitis pigmentosa, vision, disease, retina. He said, so how are you going to clone it? So we developed a method of what we call subtraction selection strategy. Subtraction was fairly new. There was only one paper before that from Mark Davis, cloned T-cell receptor a year before, using a very tough sort of method. And selection also was very hard, that was used for Duchenne gene. But we pulled out some methods using biotin. And so I proposed that and wrote my grant to NEI. I told Sherman, I want to write my own grant. He said, but you can't, you have to be in the lab. So during Christmas time, in 2 weeks, I wrote the grant. And he was not very happy that for 2 weeks I was not in the lab. And I said, that was Christmas. Holidays are for secretaries. So basically, but I did do that, and I submitted the grant and got first 1 percentile, and I became an eye geneticist. So that's sort of my story going into this. So all these mentors have been just amazing to teach me and show me the path. So this is where I focus my studies. This is how I learned along the way. If you actually, believe it or not, I didn't believe it. Almost 30% of all diseases which are listed in Mendelian inheritance of man, sort of on OMIM, that many of you probably know, they have some or the other vision component, because it's very easy to sort of diagnose those diseases. Assessment is extremely easy. If you can't see, you're going to go to the doctor. But if you're a little bit crazy, or your mind we were talking about, if you have some defects there, unless it becomes really a big problem, all of us are a little quirky. So that sort of is not very easy. But vision, you'll go. So an ophthalmologist, when they look into your eye through ophthalmoscope, this is what they see, a beautiful, beautiful pattern. And in the center, there's an area called fovea. That's where the light gets focused. Area around the fovea is called macula. This is optic nerve, which is a blind spot. The blood vessels sort of come into the eye through this, and optic nerve goes out of there to brain. So basically, that's how it looks. And even I, who I'm not a clinician, anybody can recognize that there's a problem here. This is the problem in the macular area, and this is called macular degeneration. When you have a death of photoreceptors in this area, this is what you will see. You lose your central vision, you can't drive, you can't read very easily, and there's a huge problem. And that's really the major cause of blindness in the elderly population. And then this is where the peripheral retina is sort of defective. There are all these bone spicule pigmentation that happens. And this is called retinal degeneration, which is a broad name. And there are actually literally hundreds of genes that can lead to diseases associated with vision loss in the peripheral retina. In general, that is because of the loss of initial loss of rod photoreceptors, And then in the central sort of area, macula, is generally the cone function that is defective in the beginning. So this is the structure of the retina. Very simple. It's part of the central nervous system. There's a pigment epithelium that nourishes the photoreceptors and serves as really the primary cell, which is extremely important for managing the photoreceptor function in some way. So there are two kinds of photoreceptors. These are rod and these are cone photoreceptors. In humans, rods constitute 95%. In fact, let me just put it this way. Almost 75% of all cells in the retina are photoreceptors. Of these, about 95% are rods. There are 105 million rods we have in our retina, and there are about 6 million cones that we have. Cones are distributed across, but they are much higher density in the macular region compared to the peripheral retina. But still, the number of rods is always going to be high because they're 95%. Fovea is the only region where you do not have any rods. There are only cones, and there are three kinds of cones. Short, based on the pigment that they have, short wavelength, medium wavelength, and long wavelength, or S, M, and L. In humans, you have these three cones. Only L and M cones are present in the fovea, in the center of the fovea, which is called foveola, where the highest resolution or highest visual acuity really is given through this area. Peripheral retina, of course, is very important for you to have broader field of vision. So this is what my lab, and this is what happens when you lose peripheral vision. You have central vision, but slowly even this goes away. So my lab has been focusing on that and for a long time, and this is the motivation. These are the two people. When I went to Michigan, after I got my grant, it was easy to get the job, and I had many different places. I ended up University of Michigan. This was my chair, Paul Lichter, who basically gave me all the, I was very fortunate. I had a couple of other grants that I got even before I started, and he gave me money, and I never cared about the money. I have to say that. And if I overspent, Paul was always there. This was the person, Paul Lichter, or Paul Sieving, who actually showed me that behind the tube of every DNA tube, there is a person, and I need to find those people. I need to look at those, and my work is not dependent on just the papers, but also many people depend on what we do. So this is something that, again, is the highly motivating stuff that is in my office. I show it to every student who comes to my office, who works with me, and I've had the pleasure of sort of guiding almost 500 students in my career, and many more that I've talked to. And this is from a 6-year-old girl called Amy, and her mom wrote it in, this was in Braille she wrote, and her mom sort of put it in this. And I sort of have tears every time I think about her, and I think about her probably every day, if not several times a day. Says, "Thank you for fixing my eyes. If you don't, I will still be happy, but please try." A blind girl, almost blind girl, will still be happy even though she cannot see. And and I have this bone fracture that's nothing compared to what this girl has gone through in life, but she wants us to try. She wants me and others who work there to at least try. And that's what we have been trying to do. So I'm going to tell you some very basic stuff that all of you know, but then I'm going to make it a little bit more complicated as we go along. You all know all the information that we have is in our DNA. DNA makes RNA, proteins, but in RNA has got many, many other functions as well that we didn't know earlier. Then you have proteins, everything is part of some networks. And these along the way will form different kind of cells and organs. And so basically, it's a very complicated system. There are many, many ways to do the regulation. But in the beginning, just to the basic thing that really has always fascinated me, and that's why I wanted to go to genes, recombinant DNA manipulation of those genes, it's just I want to understand how this information is transcribed or decoded to give you these phenotypes. Now, just think about it, that both healthy and disease phenotypes are coded by the same genome, and all of us are still unique, even though the information is 99.9% identical. We still have very unique features, phenotypes, functions that we do, our behavior, emotions, and everything is pretty distinct and unique, even though, but most of those variations that we have, they are in non-coding genome. And those non-coding variations, in fact, are responsible to a large extent, I would say, in changing those phenotypes, giving us the uniqueness that each of us have. All of these, this decoding of this information, DNA is same in every cell, yet each cell does different functions. They interact with each other in different ways. And all of that has to be tightly regulated. Any kind of break in that network can lead to disease. So I think basic philosophy is, if I want to know disease, if I want to really help these kids, or if I want to figure something out, I need to really understand what is normal in order to define what is abnormal. So one of the big thing, and again, I'm going to sort of give a very brief overview of what we have been doing. One of the big thing that my lab did, this is picture of a rod photoreceptor. Basically, this is the nucleus here. This is the cytosol, very small rod. And this is the inner segment, this is the outer segment. That's the functional part. That's where the light gets captured. My lab, or I, in my early days from subtraction, seeding, and cloning, when I was trying to clone the gene for X-linked RP, I also began to look at many genes, and we identified one gene that coded for a leucine zipper protein. This was in 91, and this gene I called neural retina leucine zipper gene, or NRL. And it turned out later on, I mean, I sort of speculated the reason I wanted to work on it. It seemed like a transcription factor because it had homology to, it had leucine zipper, and other leucine zipper proteins in those days were like FOS and JUN and all that. So we thought, okay, this seems very important. It turned out to be the first retinal transcription factors that was identified. So, but for a long time, we didn't know, for almost 10 years, we didn't know what it does, until this wonderful postdoc in the lab, Alan Mears. What he did was he knocked out the gene. And when he knocked this gene out, he found that in the retina, mouse retina, there were no rod photoreceptors. So as if you look at the picture, these are outer segment, inner segment, these are nuclei of the photoreceptors. All these condensed chromatin that you see in these nuclei, these are rod photoreceptors. They have very unique architecture of their chromatin. In the center, you have heterochromatin, and euchromatin, which is the active chromatin, is just in the periphery. That's unlike almost all other cells have very decondensed chromatin, just like that. They have different, heterochromatin is different parts, and these are cones. So you can recognize cones. So this is a cone, this is a cone, this is a cone. You can recognize this is a cone. You can recognize these cells very easily in the sections. And here, if you look at this, there's no condensed chromatin cell. Everything is decondensed. They're all cones. And it turns out they're short wavelength cones. They're very specific type of cones that have visual pigment, which is S-opsin. So that was a very big surprise. And Paul Sieving, who later became the director of National Eye Institute and was involved in recruiting me here 6 years later, he came here in 2001. I joined 2007 at NIH. When his lab did the ERG is this electroretinogram, it's a functional assay. And while in dark adapted ERG, you put the mice in dark and then the flashes of light. And in that, you look at rod function, because rods are extremely sensitive cells. Cones give you color vision, and they are involved in high visual acuity, and they allow you to see in the daytime, but rods allow you to see in the nighttime. So in this, you have flat ERG, knockout mice, whereas the light adapted ERG, which looks at cone function, is greatly enhanced. And that is based on, if you give the different wavelength of light, you can say that this is entirely driven by S cones. So this is fascinating. That was the first time something like that was there. But that doesn't mean that this gene is sufficient. And there are many, many transcription factors or many genes. If you knock them out, you make something else. Great. That was wonderful. This was published in Nature Genetics. Wonderful. It made my career actually sort of go faster. But then there were many other, this period was very productive for us for next 5-6 years. Another graduate student in the lab, Ado, who is now a professor in University of Nevada, what he did was, he said, if NRL is important for rod development, is it sufficient? So what he did was, he made a transgene construct and drove NRL by another promoter of a transcription factor called CRX, or cone rod homeobox gene, which was expressed just before NRL in all photoreceptors. So when he expressed NRL, this is wild type, rhodopsin marks the rod cells, cone arrestin marks the cone cells. If you now express NRL in all photoreceptors, you get only rhodopsin and no cone arrestin, means all cones are now rods. But how do you know they didn't die or anything like that? We know that because we did all kinds of different controls, and it is not. But another thing is here, NRL knockout retina, no rhodopsin, only cone arrestin. Now you put this transgene, all of these cells are now rods and there are no cones. That's the only transcription factor that I know, that when you knock it out, A becomes B, and if you put it in B, it becomes A. Sidney Brenner, so my biggest sort of source of enjoyment came when Sidney Brenner, who was my hero, if you like genes, you have to know Sidney Brenner. And Sidney gave a talk at NIH, sitting on a stool at age of 89 or whatever he was doing, and talked, half of his talk was focused on NRL and on our work, without knowing that I'm at NIH. He didn't know who the heck this guy is. But after that, when I went to him like a kid, I said, Dr. Brenner, you talked about work. Oh, I know all your work. And I was sort of like, oh my God, this is dream come true, more than any awards that you can get. And Sydney came to our lab next time and he visited NIH, and all the students and all. This was a big proud moment for me. So this was wonderful, where one transcription factor can do everything. We could use the promoter of NRL, we could turn on GFP, and we can show when exactly the rod cells are born in mouse retina. You can purify them, you can manipulate them, you can. We actually started the whole new field where you can transplant these cells to see if they can function. It didn't turn out to be as simple, but that really is a whole different story, and I don't want to talk about it. That was just wonderful again. So you can purify these cells at different stages of differentiation. You can do RNA-seq studies or microarray studies or whatever. Now later on we changed to, of course, RNA-seq, and you can see during differentiation there's a big change in even in wild type rod photoreceptor between P6 and P10. This change, as you can see, this blue and red, red is high expression, blue is low expression. So you can see these expression of these genes is going down, some of these genes are going up, and this transition really happens between P6 and P10, and that time really coincides with the time when photoreceptor maturation is taking place. So all of these things really go very, very well. Almost, and we did a lot of ChIP-seq and other studies and know thousands of genes that are controlled by NRL. And in knockout mice, many of these genes continue to be expressed and they don't turn off. And these genes that are rod specific do not get turned on. And these are S cones. And this led us to hypothesize that maybe, and these are many cone genes. This led us to hypothesize that S cones are the precursor of rods during evolution. And we had a paper in Developmental Cell where we showed that during Jurassic period, probably evolution of mammals sort of happened by development of nocturnality, and nocturnality required that you have a lot more rods. Before that, in early vertebrates, you have cone-rich retinas, but in mammals, you have a lot of the nocturnal animals have to have rod-rich retina. but we now see in birds as well, the birds, the night bird like owl and all, they have rod-rich retina, and NRL is responsible. So we propose the Evo-Devo part hypothesis that evolution of nocturnality is probably dependent on stringent regulation of NRL. In early stages, in early vertebrates, NRL is expressed in other cell types as well, whereas later it gets restricted only to rods. And this was a bunch of very good Korean postdocs in the lab, Hyunjun and all that. So anyway, so the bottom line in all of these things is that there's a long, to make a very, very long story short, these stem cells, I'm not going to mention that, but the main thing basically is from stem cells, when you're making photoreceptors, these precursors are CRX positive, which is a homeodomain protein. And if they have NRL, then these cells are going to make rods. And if NRL is not present, then they just remain as S cones, and S cone is the default pathway. And if you turn on another transcription factor called TR beta 2, then they become cones. So two transcription factor, TR beta 2 and NRL, can dictate the generation of all three photoreceptor cell types, basically. So that's the sort of big story that we can just go on digging into it and make the mechanism. Of course, mutation in NRL, many of target genes cause retinopathies. We have been identifying different domains, different residues that are associated with NRL function. And of course, it's very much dependent on, I mean, coming back to my biochemistry part, I've becoming a little bit more biochemist from molecular biology or DNA person or RNA, now going more into protein. There are a lot of post-RNA modifications that happen, and modifications of NRL allow it to interact with different sets of proteins. And this region, N-terminal region in NRL, it has intrinsic disorder. Basically, there are a lot of prolines within this region, and also serines and other amino acids which does not allow a structure. And this IRD, or intrinsically or IDR, intrinsic disorder region, is very much important for transient interactions with different sets of proteins and making of condensates during transcription. So there have been tons of advances that in the last 20, 30 years in genomics, and my lab has, in I field at least, been doing some of the things right from the beginning. We were first people to use cDNA arrays in the lab in I field, and do a lot of these Hi-C and other studies that and genetics that we have been doing. So focus currently has been basically, the questions have been, how is genetic information decoded to generate morphology and function of retinal neurons, specific photoreceptors? How do different genetic variations that I talked about, they modulate healthy and disease phenotypes? And what happens in aging and influence of environment? How is that mediated? By probably changing the epigenome, and a lot of work on DNA methylation. And can we utilize this knowledge to design therapies? So I'm going to just talk about towards the karma side. Very quickly, I'm going to go through our work on macular degeneration, which is major cause of blindness in elderly population. And again, there are three major factors. There's the genetics, but it alone cannot sort of cause disease. Even if you have all the variants associated with that, then age has to be a very important factor. It happens only in individuals over 65 years of age. And then environment, diet, lifestyle, etc., can modify the phenotype. So it's multifactorial, it's complex. Aging and environment work through epigenome, and this part is also through your genetic variations. We were part of this. We were in very early days. To again make a long story short, we had collected thousands of individuals, cases, and controls in Michigan, and then we used here. And during that period, we did some Jiva studies. On our own, we were not able to, we got some papers and some nice initial data, but the major thing happened by consortium studies where 35,000 people initially were analyzed for genome-wide association. And we got these 34 loci. This is like a Manhattan plot. Some of you may or may not have seen this. This is all the different variants and log p-value significance. And based on Banferroni correction, anything that is above 10 to the power minus 8 is significant. And you can see there are a bunch of these genes, or rather loci, I would say, that came up. Now, genome association studies are done by using mostly common variants, and these common variants, and they're just association. They are looking at the frequency in two different individuals, rather in cases than in controls. So, so it's really an association. It's not causality. So this does not allow you to say that this variant is causing the disease. Anything that is in linkage disequilibrium, very close to that particular variant, can actually be responsible. And many of these variants how do they impact gene function is also unclear. So many clinicians said, okay, you found all these different variants, different loci. Locus is really a genomic region, it's not a gene. So what does all this mean? So first we wanted to see, okay, what those variants do are in that region. So we did what we call EQTL analysis. We took over 500 human retina samples, most samples from Def Farrington, and a postdoc in my lab, Rinki, who's now at Baylor, faculty at Baylor. She really did this both genotyping and transcriptome analysis. We looked at what is called EQTLs, that is variants that can affect the function of gene. It doesn't have to be function next door, gene next door, but it could be very far away. So she looked at the variant gene relationship, and by combining the EQTL work with the GWAS, we were able to look at, using different kind of statistical tools, what gene it might be candidate causal gene for AMD. And we could do that for 6 of the loci, which was not very satisfying. We then also went to look at epigenome, because aging and environment both work by modifying the epigenome, where many of these genetic variations can affect DNA methylation, and DNA methylation can directly affect gene. And as EQTL, I was saying, variants can also directly affect gene expression. So we wanted to do the whole study, and Jayashri in the lab, working with Alet Segre at Harvard, we basically just recently published this big QTL mapping of DNA methylation and identified gene epigenome interactions. So what that tells you is that epigenome tells you the changes in the sites of methylation, which indirectly may affect gene expression. And these epigenome changes are reflecting changes because of aging, diet, environment, and many other factors. So this really, in a way, links all 3 different parts, genes with genetic sensitivity with aging and epigenome. And this allowed us to really link many other things together. So then, but again, causality is not very easy to identify. We also worked with Mike Klein at Oregon and Emily Chu. And we have many, many different large families that we analyze by both whole exome and whole genome sequencing. And we have now some big, there are over 100 families. So that's a huge amount of data that we have been working on for the last 3 years. You can only do these things at NIH. These are all multi-million-dollar studies. They're very hard to do outside. But we have now almost finished this study, and we'll be putting together some new pathways that are signaling pathways that may cause AMD. Now, coming back to that, how do those variants affect gene expression? This is just a way that's long 2-meter DNA is condensed within the nucleus. Coming back to my gene fascination with genes and gene expression. Gene expression is highly complicated process. It's not as simple. There are a lot of different enhancers everywhere, all over. And these enhancers, many of them have to be brought close to the promoter region. And so they make loops. And this looping brings this cis-regular element to close to promoter. So if you have variants in these enhancer or anywhere else in non-coding genome, that variant, if that is present in an enhancer cis element, that can then indirectly affect gene expression. That's the way to think about it. So in my lab, many different fellows, both bioinformatics people as Claire, Nivedita, Jayashri, Zach, and Jimena, what they did was they really made a high resolution map of this chromatin context using Hi-C. We combined it to many DNA methylation, histone methylation, different ATAC-seq markers, and we got the loops and we got the super enhancers. We combined all of these things to genome variants for glaucoma and AMD, and we have been able to figure out which variant might be leading to disease causality to some extent. Here is just an example I want to show you. So this was identified by GWAS. This is a lead variant in GWAS, and there are many variants in linkage disequilibrium right here. Any of these could be causal. But how it does it, we don't know. This gene is near C20ORF85, unknown gene. So that's how this locus was named in GWAS. But if you do looping studies, you know that these variants in this region, they are making these loops somewhere in this area, and they are different sets of genes. So these genes are likely to be causal, and which one, that's what we can experimentally test now, rather than genes in this region, because this can do, of course, regular expression of this, but more likely, since this is coming over here, that's what it does. And we have done a lot of chromatin architecture studies to look at that. You can do the same thing for retinal degenerative loci and find missing heritability. And yeah, and in aging, this topology of the genome is also changing. The genome is being organized to adapt to the new changes, and there's a myosin 10 gene here. And again, the topology is dictating the expression of RNA. It is reduced in mouse from 3 to 24 months. There's a loss of this looping and loss of this chromatin looping and gain of DNA methylation, which is sort of in many cases inhibits gene expression, is really leading to decrease in gene expression. So I think by combining all of these things, we're beginning to understand how disease is caused. So I want to just last couple of slides, I want to show you how all of these studies are helping. So literally we have hundreds of genes which cause retinal degeneration, dozens of genes, if not hundreds of genes that are associated with macular degeneration, and they all eventually lead to death of photoreceptors. So how does that happen? Long time ago, we proposed that there may be convergence in pathways. There may be few pathways that are really involved in all of these things, because many of these mutations may eventually be turning on one or more pathways. In a different way to think about is that everything is connected. Coming back to my early slide, everything is part of this network. In network, there are big hubs, and then there are peripheral hubs or nodes. If you break some of these nodes over here, they affect only very small tissue or cell type. If you affect the big nodes, then you disrupt the whole network, and a lot of things are affected. So if it is possible to, rather than any of these genes, each of the circle is a gene, and this is a node, any of these genes can cause the disease, probably a tissue-specific cell toxic disease. So rather than targeting each gene, can you just sort of rebuild the network by connecting with the node? Can you sort of modulate that in some way that you can set up a new equilibrium? May not be a perfect equilibrium, but can you go back to maybe 50% of function, which might be sufficient for people to see? So you can target those pathway or network. So we did some studies, and I don't want to go over once again to do that, but large screening that we did with small molecules. Here is an assay. This is organoids, and the photoreceptors are in organoids. They're labeled here. This is rhodopsin. In the control mouse organoid, you can see this is labeled with rhodopsin. This is a mutant. There are different mutations, and you can have here loss of expression of rhodopsin or reduced expression in human patient control, and this is one of the blinding disease, very little to almost nothing. So our goal is, can we screen the small molecule that can maintain this before this happens? So this is what we did, and we discovered many genes, or rather many small molecules. One of these is reserpine, and now we have more. So you can see in this patient, by treatment with reserpine, we were able to maintain photoreceptors here. In different patients, there's some, not as big as this, but we were able to do some. And now we have found other molecules of same type as reserpine, this A3, even after whatever P82, when a lot of cells have died, these molecules are able to maintain function. And now you can see here, this is in rat, so P23 heterodopsin mutation, rat. So some of these molecules can work in gene agnostic way. So this was initially identified by looking at RD16 or CEP290 mutation, but it also works with rhodopsin mutation and also another couple of other mutations. So you can see here, if you treat with reserpine or this another molecule A3, you can maintain a lot more photoreceptors and a lot of rhodopsin that you can see here. So this is sort of very exciting for us, and we are the problem in some of these cases, these are repurposing molecules, and big pharma doesn't want to get into that. So we are trying to look for partners, and these things, NIH itself can do a lot of things, but some partnership with some small biotech we are looking for right now, and hopefully somebody will. But there's a big potential because these molecules work both in organoids as well as in mice. So I want to just sort of bring back to this, how did karma came into picture? Karma came because you have genetics, your genome, these genetic variants you inherit. You have no control over these things. That's your bhagya. This is a slide I showed in my talk, and this became quite popular because you have no control over your genetics or genes. You have no control over time, Kal. That's aging. Every one of us is going to age. And we have what we have. It's very hard to control these things. This is your bhagya. This is your Kal. What you can control are your actions, and this is the epigenome part, your diet, lifestyle, and we are doing some direct evidence, rather experiments in mice directly by feeding them different diets and changing their lifestyle to see if we can control their epigenome and we can reverse some of the changes, or actually slow down, I shouldn't say reverse, can we slow down some of the changes that are happening in time and by these genes. So your actions, your karma, that is in your control, at least to some extent. So I'm going to just finish that by saying this is how I used to look like in 92. This was my lab in those days. This guy has become big CEO and all that of a company. And this is those early days. That's how it used to be. When I moved to NEI, this was our group. And this is party at our home. That's my wife here. And these are the pictures in our lab. Is very dominated by females. They are the smarter ones these days. And many of them are faculty members now. So this girl is at Stanford from Columbia. And this is from Brazil. This is from Spain. She's in a company, big executive. So there are all these different things, and it makes me very proud. This is the biggest thing in my life, of course. These are my grandkids, my granddaughter and grandson, and they're from my son. And these were when they were kids. And our daughter has a dog. And so this is a family. This is my wife. And this is, of course, my son and my daughter-in-law, grandson, granddaughter, my daughter, and my son-in-law. And this is how I used to be when I was in Bangalore. A couple of months later, I met my wife, and this is how we are now, sort of slowly after all these years. People ask me, when are you going to retire? And I say, I don't know. I have promises to keep. I have promises to Amy, and I have promises to all these. So I go to India now in Barsana, which is near Mathura, and they're all in Brindaban, and there are a lot of these kids who have. So I'm passionate about teaching, and we've started a small school there. We have no real family city or something, so we just had to choose somewhere. So this is what it is. This is the journey continues basically, and you try to take some of these kids who are living in this way towards some light, and that's where I'm going to stop, and I'm going to so my journey is still continuing, and there's sort of, as long as there is, we will, I don't know, like Professor GP, that's my inspiration when he was, when I was over there.
Thank you, Anand. That was a lot of work, tons of work you have summarized in 1 hour. Excellent piece of work, and thank you for sharing this work
Right.
with us. And
Thank you
I
so
think
much, Anand, for the wonderful talk. Very lot of data you made it so simple for people like me to understand.
Thank you.
Wonderful. Yeah.
Oh, I think, even though I never worked with you, GP, I
Yeah.
have always considered you and Gopi as two of my teachers, along with Padyati and others, because I was just fascinated by DNA, and it's just sort of craziness to the extent of leaving. I came from a family with not too much money, nothing, and denying 21,000 salary and refusing that and going to 11,000. And I tell those people that it was just sort of my craziness for genes. I just wanted to clone, and I remembered in those early days restriction sites, and I used to cram New England Biolab catalog when I was doing that. It's just and I still love to look at sequences and genes, and that's why I say how they function, what they do.
Very fascinating, Anand. The
Right.
only way I came ahead of you is I took a postdoc for 10,500. That's all I could get.
Yeah.
I was wondering.
Anand,
I was wondering what about vitamin B12?
Yeah.
High levels of vitamin B12. Do you think is better for us?
Yeah, so I think vitamins definitely have an impact in slowing down the progression of macular degeneration. And this is based on some EPID studies that are being done at NIH or NEI. My close friend Emily Chu is part of that. But the EPID effects in human populations generally are not very easy to convince people that it's really important, because there's a lot of variability in human population, right? Everybody eats different things. So generalizing something is not very, so we are trying to now do direct experiments in mice. So we are feeding, literally we are feeding. And again, as I said, you can do that. NIH is very expensive sort of thing to do. So we take mice which are 6 months or 12 months old, and we put them in different diets, and we add different things. We call them Mediterranean diet and American diet, and diet with vitamins and diet with B vitamins and blah, blah, blah. And we let them sort of go for about 6 months. And we have done one experiment. Next experiment is going to be much longer. And then there's the interaction of diet and age, right? So mice are also growing at that time. So we have to dissect that component basically within that. And we are now looking at their methylation and gene expression and everything, and trying to correlate bioinformatically how that is changing. So we know that in aging, there are changes in epigenome, in retina, in liver. So we are mostly focusing on liver and retina right now. I'm in eye institute, so of course vision and eye, but we also do liver. And we are trying to see if the changes that are happening in epigenome with time, can that change be reduced, or will that change not happen if you take a specific diet? So will vitamin, we are not working just directly B12, but we do B vitamins in general. Because the evidence is not just B12, but also maybe B6 and bunch of other.
B6, yeah, B6 also.
Yeah, B6
See,
also.
I was diagnosed with high homocysteine about 30 years ago.
Okay.
Since then, I've been taking 100 milligrams of B6 and
Yeah.
1,000 micrograms of B12 and folic acid, 5 milligrams every day.
No, I think that's all old biochemistry days from ISC that I remember we used to talk about. And I will just say the B vitamins are extremely important at our age. I think everybody should take it every day. B12, if you're vegetarian, please, I take B12 at least couple of times a week. It's very, very critical. Even when I was in ISC, I didn't realize I used to feel very tired for a couple of months. And then I went to our hospital that, on the side and here F block or whatever used to be. And they
Bravo.
gave B12 injections.
Yeah.
And I didn't realize they said, why? B12 is, no, because vegetarians have big deficiency of B12. So
Yeah.
I
I
think
am a testarian, but still I had high homocysteine.
yeah,
And,
yeah.
and there was
Anand, I have
something
a- I have a
Genetic
question.
in your case, yeah.
It
So
could be
that's-
it could be intrinsic factor deficiency also, for B12. That's why maybe I have to take it intravenous.
yeah,
Maybe.
yeah.
Yeah, Anand, I have a question. You
Sure.
mentioned reserpine. I remember before the SSRIs or serotonin receptor inhibitors came into play, the only medication we had for depression was reserpine from Rauwolfia serpentina, the alkaloid from that. So you may have a set of a population, and I wonder if there's any numbers about incidence or prevalence of macular degeneration in that population.
Yeah.
Because the serotonin reuptake inhibitors are fairly recent.
Yeah.
So there may be information available. I wonder if
Yeah.
retrospective study can be done.
Yeah. No, I actually, some of that is known. Reserpine is no longer used. It has been banned because reserpine was being taken orally for some of these things earlier, but it also inhibits dopamine, dopaminergic sort of cells.
Correct. It was non-specific.
Neurotransmission is non-specifically, but advantage in case of eye is that it's a very sort of isolated system, right? So you can inject only in the eye very tiny amount that goes to the photoreceptors. Most of it will be washed away by in the blood. It gets so diluted, the amount of that in other tissues is going to be so little that we will not have any side effects. So that was the main reason. And now what we are trying to do is we have other molecules. Some of these were anti-malaria drugs, believe it or not. Many of these are now turning out to be, they have very similar structure. And some of these can be made into eye drops. So again, the eye is such an isolated system. By putting eye drops, you have very small amount of chemical that is going into the retina. Most of it is getting washed away. And if there is some problem, you can stop eye drops. Unlike taking orally or putting genes or putting, gene therapy. So I'm not a fan of gene therapy or stem cells anymore, because those things, once they are in there, you don't know how to control them. We don't have the controlling power. But small molecules, if you can deliver by putting eye drops, you have not only control, you can control the doses, you can control the timing, you can, and none of that is going to go in your system, and in your body is going to get so much diluted that it won't have any side effects at all. So I think,
yeah, that's
yeah.
That's a good point.
Yeah.
But the main difference in the eye as opposed to other end organs is the circulation stops before the photoreceptor layer, and that layer gets oxygen and metabolites by diffusion. That's why you have retinal detachments, because there is no firm attachment to the inner layers. So I wondered, because of some of the factors you're showing, like smoking and age-related changes, is it because of stoppage of nutrients and oxygen to that layer?
Yeah, so I think you brought us a very important point. It's slightly I will change it. So retina gets, so photoreceptors are extremely vulnerable to any kind of abuse, any change. They are living at the edge. They are, in old days, Otto Warburg, we all read in biochemistry, Leninger book, Warburg effect. Warburg is aerobic glycolysis, which happens in cancer cells, and it happens in photoreceptors. So glycolysis is extremely important. So mitochondrial OXPHOS actually gives less than 50% in the mature photoreceptors. of energy. Most of it is coming from glycolysis because also it's anaerobic molecules, byproducts that are made in glycolysis that are used for this shedding, all that. So photoreceptors are extremely vulnerable. They have they're sort of chronically hypoxic, sort of in some way to say. Pigment epithelium is what gives it all the nutrients. Everything that comes, and then there's a choriocapillaris on the other side of that. So from choriocapillaris, it comes to, there's not just diffusion, there are many other transporters that are involved in pigment epithelium, which very selectively will transfer some of these things to photoreceptors. But then there's another blood circulation in the retina, which is in the plexiform layer, capillaries that I was showing in one of the early things. But that primarily gives you energy and nutrients to your inner retina. That is cells which are amacrine, bipolar, ganglion cells and all that. These ganglion cells are one that are affected in glaucoma. So much of that basically, when you say smoking part, smoking is creating smoking has got tons of other chemicals, and those chemicals are not affecting photoreceptors directly. They literally affect from blood, they affect the function of pigment epithelium. So the way I try to tell is pigment epithelium in a chess game, photoreceptors are king and RPE cells are queen. Now, king is useless if queen is not there because it cannot defend itself. Queen really sort of is extremely important to, and if a queen dies, if you get rid of that, it's not very hard to defeat the challenger. You can draw, but it's not very easy. So RPE is extremely critical for integrity of photoreceptors. And if RPE is not, so what happens in macular degeneration, the defect may come from anywhere. So smoking is a risk for macular degeneration, but defect could come from choriocapillaris, or it could be from Bruch's membrane, which is a sort of matrix in between the two, pigment epithelium, photoreceptors. The defect could be anywhere. But ultimately,
Yeah, I was sorry to interrupt, but the fovea anatomically has very few capillaries. If you, as you
Yes.
showed, most of it, it is just.
But pigment epithelium is there. Pigment
Okay.
epithelium is very unique there,
Yeah.
and it is what is still feeding. There are no capillaries there, but that is why. Fovea also has very few cells. So fovea has got a 1-to-1 ratio of cone to ganglion cells. That's why there is a very high acuity. In many other peripheral areas, maybe dozens, hundreds of photoreceptors are giving their signal only to one single ganglion cell. So it's getting information integrated from many, many areas. But in the fovea, it's just direct one cone to one bipolar to one midget or some ganglion cell down there. Basically, that's right. So it's very high acuity. So therefore, since the number is low, but pigment epithelium is there, and the pigment epithelium cells there are different from the pigment epithelium cells in the peripheral area. There are recent studies by using some beautiful AI methods are showing that based on size and based on their shape, there could be at least 5 different types of pigment epithelial cells in the human retina. That number is less in other vertebrates, just because retina is not as nicely differentiated, right? So you have, in human retina, you have many, many different areas that you can distinguish. Fovea, foveola, macula, peripheral retina, even they are different. So it's little bit, but all of these are affecting pigment epithelium. So the oxidative stress, smoking and all, once RP function goes down, then you are in trouble. And you need to really make sure those are the cells that are protecting photoreceptors.
Yeah, I have one last question. Are there populations in the world where there is very minimal incidence of macular degeneration?
Oh yeah, of course, of course. In black population, there is a less incidence of macular. So in the beginning, and even Indians, for example, a lot of people used to say they have less macular degeneration, but that is untrue because genetically Europeans are closer to us. I won't say we are closer to Europe. I'll say they are closer to us because we are more primitive population in that sense. And also the macular degeneration that happens in East Asians is slightly different in phenotype. But in Africa, the incidence of age-related macular degeneration is very low, and it's not because of age. It's not because they. So initially, when somebody used to think, oh, maybe because they don't live as long. No, but even among the older population, the incidence of AMD is much less. They have more problems with diabetes and with glaucoma.
But that would be peripheral retinopathy.
Yeah, so glaucoma is more ganglion cells basically, and that is much bigger problem there in that population. And that glaucoma incidence genetically is less, but it's pretty high still. Glaucoma is again much more complicated than probably macular degeneration because glaucoma has components coming from ocular pressure, aqueous humor. That really intraocular pressure is a big factor there, along with, but the phenotype is in ganglion cell death. So that's a sort of difference that you have.
Thank you, Anand. It's wonderful work. Thank you.
Anand, I have a question. The
Yes.
small molecules that you showed, do you know the mechanism, how they induce the
Yeah,
gene
so
expression?
initially we thought some of these molecules are turning out to affect autophagy, but now we have more evidence that they might be working through either P53 signaling or PI3-AKT signaling. So we are continuing to explore that, and we are also trying to design by AI different kinds of methods which can sort of help as well.
The small molecules that you have found, do they have any structural similarity, some pharmacophore that you can recognize?
Yeah. I think, again, I'm not a chemist, but we recruited a chemist recently, and there's a friend from India who used to work in pharma, and now he's here after retirement. So we're trying to get some help from them to look at the structures of these molecules and then see if we can design new molecules. Yes, there is a structure similarity. Again, so the good or the bad thing about me is I know where I'm weak, and if I don't know something, I try to find people who are better than I am, and involve them and give them equal credit or more credit sometimes so that they can get things. So I have got bunch of people in various areas where I'm not very good at. I just try to
Yeah, the reason I'm asking is if you can come up with a new molecule, then Big Pharma may be interested.
Yes, absolutely. So that's really where we are heading. That's exact. But even these molecules are so good that we are trying to figure out. One of my students who now runs this CRO, Ruffle, he's very excited about it. He said, your molecule seems to be, I mean, in my CRO, I look at many different molecules from pharma. Yours seems to be much better than many others I've seen. So he's going to be helping us, and we are going to try to all we need is about $10 million at this stage. So we'll try to figure out how to get it, and some of that can be done even at NIH.
You should contact Warren Buffett and Bill Gates.
Yeah, I mean, I think we need to do a little bit more. I'm a little slow, so
Okay.
I want to get some more data before I go after some of these people. But yes,
Makes sense.
because
Makes
I think ultimately you do it for these little kids and patients, and this is one of these drugs which these drugs seem to be gene agnostic. So you can have many different kinds of diseases, and I think everybody will get excited if we could show that this drug works for AMD. So we are trying to see in some system if we can have a phenotype that can be corrected by this, because AMD is a big disease. So there, big pharma doesn't get interested with millions, right? They want billions. They want a drug for AMD. They don't give a crap to some retinal degeneration, 5,000, 10,000 kids who are going blind, or 100,000 kids in the world. Many of these people are in India and Africa anyway. So, who's going to pay for that? And there's a big hoopla for, there's a gene therapy drug that just came out for $1 million. How many people in many other countries can pay for this thing, gene therapy, which works only 50% cases? So I think the idea is, if we could show that this is gene agnostic and there may be AMD kind of things associated with it, where there's even if you don't make much profit in each case, you will have a lot more. You'll still make good money. So I think the trouble is they don't care about tens of millions or hundreds of millions even. They want in billions. So it's hard to convince big pharma to spend 10, 20, 30 million dollars to make $300 million. They want to spend $30 million to make $3 billion.
Oh, hi, Anand. This is Arun. Since you
Hi.
brought up, first of all, fantastic talk. Thank you. Since you brought up gene therapy twice, I feel like you should allow me to defend it, if you will, for two main reasons. One is the very first, retinal disease was cured here at the University of Florida, and now, hundreds of patients have benefited from gene therapy. Okay, they can all see now. So, you cannot put a price tag on vision. So, these patients are extremely grateful. And I know you mentioned the cost. It's actually less than $1 million. It's $850,000, which is still very high.
150,000 less than
Right.
$9 million.
I understand. No, I understand. But the point is, FDA was convinced enough to approve it as a drug. So Luxturna was first. Yeah, go ahead.
Arun, let me not
Sure.
let you go unanswered, because I've been
Okay.
following. I'm in the field and I've been following. I know
No, I know.
thing, and I know where it began in Jean Bennett and at University of Pennsylvania, where it all started. So they
Well,
have been.
sorry, I have to interrupt you. Sorry, it started actually here with Bill Houseworth, and then
No,
Jean Bennett took over. Yeah.
Bill Houseworth was part of the team that Jean Bennett. So Bill
No,
Houseworth.
it's the other way around. Actually, Jean Bennett was the part.
Okay. Bill, okay, don't let me go to Bill, because I know Bill probably for a very long time. He takes stuff from too many. So he used some of our vectors and never gave us even credit for that. So Bill is
Oh,
Bill.
sorry to hear that.
Yeah. So no, but the thing is, this mouse model and the humans stuff and all that. There have been a lot of groups. This is not one person's stuff. This is really a team effort, which was funded by NIH and then FFB before other things happened. So it doesn't matter what happened. But the bottom line, I think, in all of these cases is that there are now big reports that suggest that only 30% of the kids are fully getting benefit. 30% it doesn't even work very well, and you have to really treat it very early when these kids are young. If you do it in later stages, it doesn't even work very well. So it's not really as simple, and it works only for RPE65, just because that's a pigment epithelium cell and not a neuron, and it's easier to do that. Since then, there have been 250 different trials in using gene therapy. Nothing has come out in market, and maybe some will come out after spending billions of dollars. So I've been following this field, Arun. I know you are a gene therapy person, but the bottom line is retinoschishistrel, which the director here really spent millions of dollars. And there are many companies. Some may work. I'm not saying it doesn't work. All I'm trying to say is that billions have been spent by dozens of companies, and many of them, big pharma is now backing out, because they know Roche is having second thought of after buying Spark. So Spark, which is what is credited with this RP6 or Luxturna stuff, they were bought for $4 billion.
Yes.
Okay, and Roche, and a lot of scientists and other people at Roche told them not to do that, but they did, and now they're having buyer's remorse basically, and a lot. Novartis just closed down all their opti thing. They spent 100 million plus, and nothing came out, and everybody was focusing on. I was being recruited for gene therapy by one of those companies. I said, "What can you do for me?" They said, "They'll double my salary." I said, "What else? Will you listen to me? 50% I want to do my own stuff." They said, "Forget it." I said, "You forget it. I don't care." The thing is, the bottom line is that's one of the things that you should try, but people have abandoned it just because it looks very sexy. Gene therapy is sexy. I mean, I agree with that. The gene editing, how can you using CRISPR? It's all very sexy stuff. At the end of the day, can you take it to public? No. And can you take it to India, Africa, China, or many other places? No. Yes, in US, the system is going to go bankrupt tomorrow if everybody starts charging 850,000 for every patient. So
Okay, so will you now allow me to defend myself? Just
Sure,
the point that I
absolutely.
make. So I completely agree with everything that you said. The problem has been, and I've talked about this at this meeting. So, again, I always give my talk, and I always, show a picture of AAV and, make this comment that AAV evolved as a virus, not as a vector. Okay. So every, company that is, pursuing gene therapy is using the first generation of AAV vectors, which is clearly extremely suboptimal. There's no way the human immune system will not take care of, just like it takes care of the first, the initial, the naturally occurring virus. So the vector has to be different from the virus. And this is what we have been pursuing. So we have generated the next generation of vectors. They completely evades the immune system and much more efficient. And therefore, you cuts down on the production cost, it cuts down on the dose, and therefore it's going to be much more effective. So we have a trial actually about to start in India, which I've actually briefly talked about at this meeting. I think that is the answer. So, yeah, you cannot take naturally occurring virus, which is a very inefficient virus to start with, and begin to expect it to perform miracles. That will never
Yeah.
happen.
No,
So we have, yeah. So that's what we've been pursuing. And just one last comment. I think I apologize because I'm not in this field, but we have beyond capsid modification, which is the next generation vectors we have created. We have since been pursuing genome modifications. Remember, AV genome is single-stranded, and it is a very, very inefficient genome because there is no RNA polymerase that can transcribe a single-stranded DNA genome. So we are focusing on modifying the ITRs, which is the inverted terminal repeats of the AV genome. And it's interesting that you talked about. I know I should have known this, but I didn't. So we are actually creating a retina-specific ITR. So we actually took your NRL sequence and inserted into the AV ITRs, so to make it retina-specific, highly expressing transgene products.
Yeah.
So we really have to modify not only the capsid, but also the genome to make it more efficient, less immunogenic, and less expensive, to make it available to a large patient population outside the US and Europe, especially in the developing world. So I don't see that, gene therapy, I don't call it sexy. I think it's a very practical solution to a large group of population, which will otherwise never benefit.
So Arun, let me clarify a little
Yes.
bit more. What happens in science in general, and I'm talking from retina perspective, but it probably happens in general terms. When something goes through one round, suddenly it becomes a fad and everybody starts doing it. And what happened in NEI, particularly from director who used to be a big fan of gene therapy, all the way down to everybody else, they just go after one thing. Now, science, in my mind, is much broader, and you need to really put your hand in everything because you don't know where what is going to lead to some new discoveries. So what happened is all the money was going to gene therapy in the beginning, and then stem cell, where we were part of in the beginning. But I realized long time ago that this transplantation, the way it's being done, is not going to work. I pulled out, and I tried to tell people. They didn't, and of course 6 years later they found out the way it's being done is not. So all the money was going to stem cell and transplantation for some time. So now the shift is oh, small molecules. There are a lot of people are beginning to do now small. So it's just, in a way, it's crazy stuff. Good thing has to be that you do a little bit of this, little bit of this, little bit of this. You really try to bring everything. In my mind, everything is important. You can't really focus on one thing. And I have strong feelings when people just go after just one thing. I'm not talking about individuals. I'm talking about the policy. I'm being at NIH and being in NEI. I am involved in some of the things and some foci, and that's what bothers me all the time. They just go crazy after one thing without realizing that everything you have to continue to do. Maybe you go a little bit more focus on one than other. So of course, in I field also, the many people, I'm sure you know, John Flannery and Denise Delcara and others who have been also trying to modify capsid and other things. And of course AAV is important. Of course this is going to be the new for gene delivery. I'm more than gene therapy. I'm going to call that a delivery vector. And I'm going to say what you and others are doing is amazing, but you don't have to do gene replacement or one gene at a time or anything. Won't it be wonderful if you define a gene or a protein which can target maybe 10 different diseases, so that in a gene agnostic way, you can do that by small molecule, but you can also do by delivering a gene within a network which affects many, many different parts. So I'm not saying that this is good or bad. All I was sort of reacting to is that such a high focus, literally billions of dollars went in one direction, and other parts of research were not getting funded because the director and other people like certain kinds of things. And then they, no, you need to just sort of spread your, don't put all your eggs in one basket, right?
I completely
Anand,
agree.
I
Yeah.
think that is the history of the science, man. Any new
Yeah.
technology comes, there is always a rush of money. And good part is that that rush of money tests the utility of the technology, how far you can take it. If
Yeah.
it doesn't have the power that it promises, it will fall off.
Yeah.
And something else will come. So that is the history. Even for the small molecules, if you go back 50 years ago when this high throughput screening started, people put billions of pharma billions of money on that effort, and a lot of things got wasted. So it's a learning lesson. There is nothing one way to do things. There are several ways you can skin a cat. So there is gene therapy, there is small molecule, there is CRISPR. There are different things, and some of them work for some of them don't work for them. So there are different approaches.
Yeah.
Yeah, I completely agree. There's no, um, one approach that is going to be successful. It has to be a combination of approaches. And I don't disagree with you at all. I'm just saying that, these people otherwise had no options. I mean, these are, very rare diseases. Something had to be done. So
No,
obviously
I
this is
agree. I agree, but I'm more on a public health perspective.
Yeah.
These kids are not the only one. There are thousands of other kids, and
Sure.
how do we reach that? And I think I was
Yeah.
talking more of a general perspective, and I fully agree that. But I'm I also sort of. Um, I always fight about in NIH and other places about basic science, because there's this huge rush for this so-called translational thing in basic in funding and all that, because all of this came because we funded basic research as well. That's why we are leaders in the world, because we understood things at the basic level, and we were not thinking where they are going to go, but they did. And I think we can't stop that, and we can't just try to put everything over there. So I'm more from that perspective. Wherever I get a chance to
Yeah.
talk
But
about it.
Anand, it is the translation and research that eventually funds the basic research in a way,
But
Just
yes
basic
and no.
research is okay, but you have to make it practically utilizable.
I would disagree with that. And NIH for research was funded by not it's such a small amount of money overall that still goes to NIH. And the things that have come out of research funded by NIH are just enormous. Industry basically feeds on that. Industry does not put much research money on basic research. Industry feeds on that.
Yeah, so this has been actually studied in detail. Every dollar that the NIH spends, the return on investment is $7. So you can
Exactly.
imagine. So yeah, clearly basic science is absolutely needed, and we are losing sight of that,
Yeah.
no pun intended. So but again, I'll make one last point, which I hope you will agree with. The first 50 years of discovery of AAV, there was zero interest. No one was interested in working on AAV because it doesn't cause any disease.
Yeah.
So much so that what AAV used to be called? Almost a virus. They say
Yeah,
it's not
I think you mentioned it in the
right,
previous talk.
exactly. It's not a real virus, doesn't cause any disease, doesn't do anything. So hardly anyone, was even pursuing any research on AAV. So it was almost impossible to get funding from the NIH. Even NIH was not interested.
Yeah.
And so since then, with all the caveats of the first generation vectors, we still think that AAV, has a place in not only in basic science but also its application for helping patients who have otherwise. I will just give you one example. The disease called I'm sure you're aware of this, spinal muscular atrophy.
Yeah,
It's
yeah.
absolutely a death sentence. These kids are born completely paralyzed. Average life expectancy is 1 year. Now
Yeah.
these kids are 5 or 6 years old. They're thriving, and single injection has cured this disease.
Yeah.
So yes, I agree that it's to use your term, it is gene therapy is sexy, but talk to the parents, whose kids are suffering from these deadly diseases. They will do anything to help their kids, just like you and I would do.
Yeah.
So yes, this is a growing field, and hopefully things will get better and cheaper. And
No, I agree with you 110%. The AAV example that you gave, and I was going to give that to you, but I'm sure you knew that anyway. So that's what you're saying. That this is what I say basic research. This is where it began. Looking at people ask me, why do you? So somebody commented in one of the grant proposals when I used to be there, one of my colleague who didn't get funded, who was doing crystal structure of this. Why do we want to do that? We know this, the NRL gene, everything, and why do you want to get Crystal? Anand, your collaborator Anand has done already everything. I said, you are so stupid. You're so stupid. Whoever the reviewer is. So I had to tell my friend how to write to see you can, you talk about personalized medicine, you talk about molecule, you talk about how you can manipulate in precise manner, what you can do, where you can target. What are you talking about? The crystal structure is not important.
Yeah.
So I think sometimes people just go in such a direction without really carefully thinking. To me, everything is important.
absolutely.
Of course, some
Absolutely.
things are a little bit more important than others at any
Yes.
given time. But to deny that, to say that, oh, we should all go in this direction, that direction, that's really where I was coming at.
I
I fully
agree.
agree with you, Arun. Yeah,
Yes, likewise.
yeah, yeah.
Thank you very much. It
Great,
was
guys. Wonderful,
illuminating.
wonderful discussion. So
Fantastic. Thank you.
this is really
Jagmohan Singh
exciting.
is
Good to
joining
know.
now. Once everything is all over, we were going to get say goodbye and hi Jagmohan. Maybe he
Thank
got a
you.
little late. Still,
Yeah, he's in Canada. He got.
yeah, he's in Canada. Yeah. All
CK, you're
right.
muted.
Thank you for a very nice talk, and we enjoyed and lovely discussions.
Thank you.
Hope
Thank you.
such meetings will continue.
Thank
Yeah.
you. Thanks.
Bye.
Wonderful
Jeff
seeing you,
and
Anand.
Michigan.
Take
Yeah,
care.
great seeing you. No, I have to start joining more and more often. This is
Yeah.
fun to see you guys. Fun to
Yeah.
see all the faces. Yeah.
See the kind of
Thank you.
discussions we have, right? Very exciting, right? You should come.
Yeah, it is. It is exciting. Hi, Krishna. But how are you?
Very nice discussion, the presentation. It was very educative. Thank you so much. Thank you.
Thank you. Thank you.
All right.
Thank you. No, I keep
Bye,
reading
guys.
about all that.
I
I
gotta
read
go.
all
Bye.
the
Mhm.
Yeah,
See you
I read
later.
all the emails. It's so much fun to read those emails and get perspective and have the recorded conversation sometimes. Yeah.
Okay, I should stop
All
the
right.
recording.
All
Yeah.
right.
Thank
Thank
you.
you.
Thank you.
Okay,
Thank you, Anand. Thank you.
Bye.
Bye.
Good to see you again. Chandru, I see more often.
Yeah.